Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome back, everybody, for another deep dive. This time, we're diving into the world of irregular heartbeats.
Sounds exciting.
It is. I think you'll find it pretty interesting. We're looking at long QT syndrome.
LQTS.
LQTS, yeah. You know, when I first heard about this, I always sort of thought it was just one condition.
Right.
But we're going to learn today that it's probably not. So we're basing this deep dive on some research that came out of Vanderbilt University recently.
Sounds good.
So let's start off. Maybe you can just explain for everyone, what is LQTS?
So LQTS is a heart condition where people have irregular heartbeats. And this is because there's a problem with the potassium channels in the heart muscle cells.
Okay, so when you say potassium channels, what do those do?
They are very important for regulating the heart's rhythm. They essentially act as a reset button for the heart's electrical activity after each beat.
Like a reset button?
Yeah.
Okay.
So in LQTS, this reset process is disrupted, and this leads to the irregular heartbeats.
Okay. So is this a common thing?
Well, there are many people with LQTS, and there's a specific gene responsible for these potassium channels, the KCNQ1 gene.
Okay.
And what makes LQTS really complicated is that this gene has over 2,500 documented mutations.
Wow. So how are you supposed to figure out what's causing it if there's so many different possibilities?
Right, and that's exactly where the research from Vanderbilt is so helpful.
Interesting.
It can be difficult to pinpoint the exact cause, and then that makes it even harder to find the best treatment for each patient.
Yeah. So 2,500, that's a lot of mutations.
Yeah.
So tell us a little bit more about the Vanderbilt research. Why are we talking about it?
Well, they took a really in-depth look. They analyzed 61 different KCNQ1 variants in their study, which was recently published in TNAS.
Okay.
And what's really unique is that they didn't just look at whether or not these variants cause disease. They really dug deeper to understand the specific mechanisms at play.
So the how, not just the what.
Yes, exactly. They used some advanced techniques like flow cytometry and electrophysiology, and that let them get a really detailed picture of how each variant actually affected the potassium channels.
So I'm trying to picture this. What is flow cytometry?
Well, that's a technique that allows researchers to track if the potassium channel protein actually reaches the surface of the cell.
Okay.
Which is super important because if the channel is not on the cell's surface, it can't function properly.
Okay, so it has to actually get there.
Exactly.
And electrophysiology, what's that do?
So that allows us to measure the electrical currents flowing through these channels.
Okay.
And that tells us, are they working correctly or not?
Oh, okay. So it's like, is it actually working or not?
Exactly.
And was it all like just a simple case of, like, the channel doesn't work?
Actually, no. That's what makes this research so interesting.
Okay.
What they found is that there wasn't just one mechanism at play, but they identified 5 distinct classes of KCNQ1 variants. Yeah, and each of them has a unique way of disrupting the heart rhythm.
So it's not just LQTS. It's like 5 different conditions, potentially, that all have been called LQTS.
Exactly.
Oh, that's really interesting.
It really challenges our understanding of the condition, and I think it really opens the door for more personalized treatment.
Yeah, because if it can be 5 different things going wrong, it can't be one treatment that's going to fix it, right?
Exactly. So, for instance, some variants were unstable and couldn't even reach the cell surface, but others were stable but just had reduced activity.
Oh, so it got to where it was supposed to go, but it just wasn't really doing its job.
Yeah, and in some cases, they found that variants functioned completely normally.
Really?
Yeah.
So it's possible some of the variants that they thought were causing the problem are just, along for the ride.
That's a real possibility, and they could be totally benign.
Wow. Well, this has really blown my mind. So obviously this has huge implications for how we diagnose and treat LQTS.
Absolutely. It shows that we need to look beyond just identifying a KCNQ1 variant.
Okay.
We really have to understand the specific mechanism behind each patient's condition.
So that's what everyone's talking about when they talk about personalized medicine.
It's a big part of it.
Okay, so what would that look like? I mean, I hear this term personalized medicine all the time, but I don't really think I understand it.
So imagine we could analyze a patient's genes and we pinpoint which of these 5 LQTS classes their variant falls into.
Right.
And then we can tailor a treatment that targets that specific mechanism.
Right.
Instead of just a one-size-fits-all approach, we're basically customizing the therapy.
So it's like knowing the exact problem and
Okay.
giving the exact solution.
Precisely.
That's really cool. So what would that look like? Give me an example.
Well, one patient might need a drug that helps stabilize their potassium channels, while another might benefit from a drug that boosts the channel's activity.
Oh, I see. Okay. So it's like some people need more, some people need less. It just depends on what their particular genetic makeup is.
Exactly. And this approach, it's already being explored. There are various labs at Vanderbilt and beyond looking at this.
It seems like this is really the future, this sort of personalized approach.
Yeah.
But there's another part to this, right? Like we have all these tools now that are supposed to predict, like if you have a certain gene, does that mean you're going to have a certain disease?
Right.
So did they look at any of that?
They did. That was another important aspect of this research. They actually put 8 different disease prediction algorithms to the test.
Oh, wow.
They compared the predictions that these algorithms made against their experimental data.
So how'd they do?
They showed some promise, but there's definitely still room for improvement.
Okay.
The algorithms were generally good at predicting disease caused by those misfolded proteins.
Okay, so the ones that just didn't, form right.
Right, but they struggled a lot more when it came to variants that were affected by more subtle changes, like those changes in the electrical signals.
So it's like, if it's a really big glaring error, they can find it, but if it's something kind of more subtle, then they have a harder time.
Yeah, exactly. It's interesting, though, because Stanford's Revel algorithm had the highest prediction accuracy
Oh, okay.
at 85%.
That's pretty good.
It is pretty good. But even AlphaMissense, which is a tool developed by Google DeepMind related to AlphaFold 2, wasn't among the top performers.
Oh, that's surprising. I thought those were supposed to be the best of the best.
I know, but I think it just goes to show that there's still a need for more research, especially in this area.
Yeah, we still got a lot to learn.
We do. This study highlights the potential of these algorithms, but it also shows that there's a need for continued refinement and development.
That's so interesting. It's like we've come so far, but we've still got so far to go.
Absolutely. Imagine a future where we could not only predict if a variant is pathogenic, but also the specific mechanism behind it.
That would be game-changing.
Yeah, and even better accuracy than we have now.
Wow. Well, this is all really fascinating stuff. So to recap, what have we learned so far? I know I've learned a lot.
Yeah, me too.
So we've learned that LQTS is way more complex than we thought.
Yeah, definitely. It's not just one condition, but 5 distinct classes that are all driven by different mechanisms.
And that means, of course, that personalized medicine is going to be key to treating this.
Absolutely. We need to tailor those treatments to each patient's unique genetic makeup.
And those predictive algorithms, they're showing promise.
They are, but there's still room for improvement when it comes to accurately diagnosing LQTS based just on a person's genes.
Okay. Well, this has been a fantastic deep dive so far. We've still got more to talk about.
I know, there's so much more.
So stay tuned for part 2 of this deep dive, where we're going to be talking about the future of LQTS research and what all this means for personalized medicine and cardiology as a whole. And we are back for part 2 of our deep dive into the world of irregular heartbeats and personalized medicine.
Yeah, we're just getting started.
I know, there's so much more to unpack here. So one of the things that I thought was really cool about this research is just how collaborative it was.
Yeah.
It wasn't just one lab working by themselves. This involved researchers from all different institutions, each bringing their own, expertise to the table.
Collaboration is key.
Yeah.
When it comes to science.
Especially, with a problem this complex, you need a lot of smart people working together.
Exactly. And I think in this case, you have Professor Charles Sanders at Vanderbilt and Dr. Al George at Northwestern. And they really combined their deep knowledge of biochemistry and pharmacology, electrophysiology, all these things coming together to really dissect the issue.
Yeah. And it wasn't even just the senior researchers.
Yeah.
Professor Sanders specifically called out the grad students
Yeah.
who were involved in the research.
That's great.
Catherine Butcher and Carlos Inouye, he was just singing their praises, talking about how much work they put into this.
Yeah, that's really important, mentorship. Training the next generation of scientists is so important to moving any field forward.
Yeah, definitely. So let's talk a little bit more about the big picture here. Personalized medicine.
Right.
We kind of touched on this earlier, but I want to go a little bit deeper. I want to understand, what could this future look like?
Well, this study really just, highlights how understanding the specific genetic underpinnings of a disease can lead to much more targeted and effective treatments.
It's like we're moving away from just treating the symptoms to actually understanding the root cause of the problem.
Yeah. It's like a roadmap for each patient,
Yeah.
guiding us to the appropriate therapy based on their genetic makeup.
Okay, I like that, the roadmap analogy. And this research obviously was focused on LQTS.
Right.
But the implications of this go way beyond just this one condition.
Oh, yeah, for sure. The principles can be applied to so many conditions, from cancer to neurological disorders, really anything that has a genetic component.
So it's like moving away from this one-size-fits-all approach to medicine.
Exactly. Embrace the complexity and the individuality of each patient.
I mean, it's super exciting.
Right.
Like, the possibilities are just, like, endless. But, of course, anytime you have a new technology, you have to think about the ethical considerations.
For sure.
What are some of those? What are some things that we have to be mindful of as we, delve deeper into this personalized medicine world?
Well, privacy is huge. Data security.
Uh-huh.
And then the access to these potentially life-changing treatments.
Yeah.
So, like, how do we make sure that genetic information is used responsibly?
Yeah, like you don't want to get your genes sequenced and then suddenly no one will give you health insurance or, something like that.
Exactly. We need strong regulations and safeguards in place to protect individuals' genetic privacy.
Right. And the access question is huge, too, because, a lot of these new technologies, they're really expensive.
Yep.
And so how do we make sure that personalized medicine is actually benefiting everyone, not just people who can afford it?
Right. We don't want to exacerbate existing healthcare disparities. We want to promote equity and make sure these treatments are accessible for all.
So these are big questions, and I don't think they're easy answers, but they're questions that we have to be talking about.
We definitely do. And I think it needs to be a conversation between scientists, policymakers, ethicists, and the public.
Yeah. So this deep dive, I mean, this has just been a whirlwind of information.
No.
So much to think about. We learned that LQTS is more complicated than we ever thought.
Yep.
We've talked about personalized treatment, the ethical considerations, but that is not all. Stay tuned for part 3, where we're going to go even deeper and explore some other really interesting things that, like, spin off of this research. Things like protein folding and the role of AI in medicine and what all of this means for the future of cardiology.
And we are back for the final part of our deep dive into the world of irregular heartbeats.
It's amazing how much ground this one research article has allowed us to cover.
It really is. It's like a jumping off point.
It is. Like, it just opens up all these other avenues for exploration.
Exactly.
So one of the things that I found really interesting, and we talked about this a little bit in the last part, was this whole idea of how your particular genes, like the actual makeup of your genes, can really influence the course of treatment for something like LQTS. But one of the things I thought was really interesting was this mention of protein folding. Can you explain that a little bit? Like, what is that and why is it important?
Sure. So these potassium channels that we've been talking about, those are proteins. And proteins, they're like the building blocks of our cells. And the way that they're folded, their three-dimensional structure, that determines how they interact with other molecules in the cell.
So it's like how it's shaped determines how it works.
Exactly. You can almost think of it like a key fitting into a lock.
Okay.
If the key is bent out of shape, it's not going to work.
Right.
And similarly, if a protein is misfolded, it won't function properly.
Okay, so it has to be like the right shape to do its job.
Exactly.
And this research showed that some of these KCNQ1 variants, some of them were actually prone to misfolding.
Yes.
Yeah.
And that meant they couldn't get to where they were supposed to be on the cell surface.
Right, so they're like the wrong shape, so they can't even get to their job.
Right.
Wow. So is this something that's just like specific to LQTS, or is this something that's important for like a lot of different diseases?
Oh, no. Protein folding is a really fundamental process. It's important for tons of diseases. So understanding how it works is really important for like developing new treatments.
Okay, so this actually kind of brings us to another really interesting topic that I wanted to touch on, which is artificial intelligence. Because AI is being used now to try to understand and predict protein folding. We mentioned AlphaFold 2 a little bit earlier.
Right.
Can you tell us a little bit more about that? Like, how is AI actually being used in this field?
So AlphaFold 2 was developed by Google DeepMind, and it's been incredibly successful at predicting protein structures based just on the amino acid sequence.
Wow.
And this is a huge deal. Like, it's really revolutionizing our understanding of protein folding.
And so how does that tie back into like the disease aspect?
Well, if we can use AI to figure out which genetic variants are likely to lead to misfolded proteins,
Right.
then we can start to predict which variants are likely to cause disease.
So it's like having this like super powerful tool that can just like sort through mountains of data and be like, okay, this one's bad, this one's not so bad.
Exactly.
That's incredible. So we've talked a lot about LQTS, this irregular heartbeat. But zoom out a little bit. What does all of this mean for, like, the future of cardiology research?
Right.
Where do we go from here?
I think it opens up so many possibilities for personalizing treatment for a whole range of heart conditions, because a lot of cardiovascular diseases, they have a genetic component.
Right.
And if we can understand those genetic links, we can do things like diagnose people earlier, maybe even come up with preventative measures.
So it's like, instead of waiting until someone's already sick, you can be like, okay, we know you're at risk for this. Let's do something now to try to prevent it.
Exactly. Imagine being able to look at someone's genes and say, okay, you're at high risk for heart disease. And then tailor lifestyle recommendations, maybe medication, or even gene therapies to try to prevent it,
Wow.
or at least delay it.
That's incredible. It's like moving away from this reactive approach to healthcare to something that's much more proactive and personalized.
Exactly.
Wow. Well, this has been an amazing deep dive. I feel like we've learned so much. We started with LQTS, this irregular heartbeat.
Right.
And we've talked about personalized medicine and the ethical considerations, protein folding, AI. It's just been incredible.
It's really shown how all these different fields are all interconnected.
It really has. And so to wrap things up, I'm going to leave our listeners with a question to think about. If we can personalize treatment for irregular heartbeats based on these tiny variations in our genes, what other areas of medicine could benefit from this same approach? The possibilities really do seem endless. Thanks for joining us on this deep dive. We hope you'll continue to explore the fascinating world of science and medicine with us. Until next time.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.